You know, when you look at the chips inside, well, everything these days, your phone, your computer, the cloud, it's pretty amazing what we can do.
It really is breathtaking engineering.
Yeah.
Truly.
But there's this like fundamental limit we're bumping up against. Everything has to be built flat.
Yeah, it's like we're trying to build this incredibly complex, dense city, but we're only allowed to build bungalows, no verticality.
Exactly. And for the next generation of stuff, think quantum computing, really advanced AI, super sensitive sensors. We need that vertical dimension. We need like the Coruscant cityscape from Star Wars, not just a flat grid.
That's the goal. Real 3D architecture.
Yeah.
Down at the nanoscale. But our standard tools, lithography,
Yeah.
because basically like super precise carving.
Right.
It just doesn't cut it when you try to make things smaller than, say, 50 nanometers and build them upwards.
Yeah.
It gets messy, unreliable.
The physics starts working against you, right? The top-down methods where you carve away material, you just hit a wall.
They really do. Heat, weird quantum effects like tunneling.
Yeah.
It all becomes a massive problem in those flat stacks. So, yeah, we need a totally different playbook.
And that's what we're diving into today. We're looking at this really radical new approach that kind of throws out the chisel and hammer and picks up the ultimate nanoscale builder instead.
DNA.
Exactly.
Yeah.
We're looking at sources talking about this, specifically a paper in Science Advances. It's called Scalable Fabrication of Chip-Integrated 3D Nanostructured Electronic Devices via DNA Programmable Assembly.
It's a big deal because it's about merging biology's precision, DNA, with our standard silicon chip-making processes.
Okay, so let's unpack that. You mentioned top-down earlier. That's the carving method.
This is just subtractive. You start with a block of silicon and etch features into it. It built Silicon Valley. It's powerful, but it struggles below 50 nanometers, and it's really bad at making complex, porous 3D shapes.
You can't really carve a sponge at that scale.
You absolutely cannot. So the alternative is bottom-up.
Right.
Building things molecule by molecule, letting them self-assemble based on their inherent properties.
And that's where DNA comes in. Not for genetics, right? But for its structure.
Precisely. DNA strands have this incredible specificity. A only pairs with T, C only with G. It's like molecular Legos that only fit together in very specific programmed ways.
The ultimate self-assembling toolkit.
And the authors of this paper, folks like Aaron Michelson, Lior Shani, and the lead researchers, Vlad Pribiak at University of Minnesota and Oleg Gang at Columbia and Brookhaven National Lab, they harness this using something called DNA origami.
DNA origami. Okay, that sounds cool. How does that work?
It's brilliant, actually. You design long strands of DNA and shorter staple strands that force the long strand to fold into a specific 3D shape you want. In this case, they made tiny octahedrons, like little 8-sided diamonds, only about 29 nanometers across.
Wow. So these are the basic 3D building blocks, the tiny bricks for their nanostructure?
Exactly. They call them material voxels. But here's the catch that tripped up earlier research.
There's always a catch.
Right. People could make these cool DNA shapes in a test tube, loads of them. But then how do you get them onto a chip where you need them? Usually, they just pour the solution onto the chip, drop casting.
Which sounds random, and random is the enemy of manufacturing, right? You need structure A here, structure B exactly 2 micrometers over there.
Precisely. You can't build reliable circuits with random placement. That's the key problem they solved. They figured out how to bridge the gap between the messy, bottom-up self-assembly and the precise top-down world of chip making.
The hybrid approach. Okay, how did they make that connection? How did they tell the DNA where to build?
Super clever. They used the standard top-down lithography first. They patterned tiny squares of gold onto the silicon wafer, exactly where they wanted the structures to grow. Think of them as landing pads.
Okay, so the chip has designated spots marked with gold.
Then they attached special single strands of DNA, ssDNA, only onto those gold pads. These strands act like molecular Velcro or specific anchor points.
So the gold pad says, build here. And the specific DNA strand attached says, only this kind of structure can dock here.
You got it. These anchors make sure the 3D DNA origami structures only start growing on those designated gold squares. It's called selective growth. And they got it to work over large areas, like millimeter scales.
And it worked well. Like, how precisely could they place them?
Amazingly well. The paper mentions like up to 97% of the little 5-micron gold pads got covered. And over 80% had just a single, perfectly formed crystal structure on them. That's getting close to manufacturing reliability.
That is impressive control. Just getting them in the right place is huge.
It is. But here's where it gets really wild. The wow factor moment for me, anyway.
Okay, lay it on us.
They didn't just control where the crystals grew on the chip. They figured out how to control how they grew vertically, their orientation.
Wait, like controlling the crystal shape just by changing the DNA anchors?
Yeah, basically programming the z-axis, the upward growth. They found that by changing which specific DNA anchor strands they put on the gold pad, they called them X and X prime, they could completely change the crystal's orientation.
Okay, how? What happened if they used just one type, like only X strands?
If they use just X or just X prime, the crystal grew in what's called the 111 direction. Picture it growing outwards and upwards, forming like a pyramid shape. This maximizes the surface area exposed.
Okay, a pyramid. Good for sensors, maybe. Lots of surface.
Exactly. Great for things that need to interact with the environment, like gas sensors. It's a specific crystal shape known as a winter-bottom morphology.
Okay. But what if they put both X and X prime strands on the gold pad?
Total change. The growth switched to the 100 direction. It grew straight up, forming a more cubic structure, like building a little tower instead of a pyramid.
Wow, so just by tweaking the DNA anchors, they switched between building a pyramid and building a cube.
Right. From optimizing surface area to optimizing vertical stacking. That's huge for designing different kinds of devices, maybe stacking memory vertically or building those complex neuromorphic circuits.
That is incredible control. But, okay, practical question time. DNA is squishy, right? It's biology. How does that survive on a chip? Chips get hot. There are chemicals involved in manufacturing.
Totally valid point. You can't run a processor on a scaffold made of Jell-O, basically.
Mm.
The DNA provides the blueprint, the precise shape, but it's not the final material.
So they have to solidify it somehow.
Exactly. They needed a way to turn the soft DNA structure into hard tech. It's a two-step conversion process. First, they infuse the DNA structure with silica using a sol-gel process. It forms a perfect silica replica, preserving every detail of the DNA framework.
Okay, so now it's a hard silica structure like glass, but shaped by the DNA.
Right. Stable, durable, but silica is an insulator mostly. Not great for electronics.
Need a semiconductor. Precisely. So step 2. They take that porous silica framework and infiltrate it with a functional material. In this case, they used tin oxide, SNOX, which is a semiconductor. They used a technique called vapor phase infiltration, VPI. So like flowing a special gas through the silica sponge, and it deposits tin oxide everywhere.
That's a good way to think about it. It coats the entire internal silica scaffold, so you end up with a 3D nanostructure of tin oxide, perfectly templated by the original DNA origami shape.
And did they check if it worked? Did the final structure look right?
Oh yeah, they use powerful microscopy, STEM, and X-ray techniques. They confirmed they had these tiny, like 2-nanometer tin oxide crystallites forming that precise 3D lattice they designed with the DNA. The blueprint was successfully transferred to a functional material.
Okay, the ultimate test then. Did it actually do anything? Did it work as an electronic device?
It absolutely did. They hooked up electrodes to these 3D tin oxide structures and shone light on them, specifically UV light. And they saw a significant jump in electrical conductivity, a clear photocurrent response. When the light was on, current flowed much more easily, around 300 microamps. Turn the light off, the current dropped right back down.
So they built a working nanoscale light detector using DNA as the architect.
Essentially, yes. A highly sensitive one, too. And the way it responded to light followed a model, the Poole-Frankel model, which is typical for materials with lots of defects, or importantly, lots of surface area within a structure.
Ah, so that porous 3D structure they built using DNA wasn't just a structure, it was actually key to making the device work better.
Exactly. That huge internal surface area created by the DNA template seems to be really effective for generating and transporting charge when hit by light. It validates the whole approach. The DNA didn't just build something, it built something optimized for the function.
That's fantastic. They went from DNA strands to a working electronic component with programmable structure.
It's a major proof of concept. It shows this hybrid top-down meets bottom-up approach isn't just theory. It can make real, functional, complex devices that we couldn't make before.
Okay, let's try and bring this home for everyone listening. What are the big takeaways from this work out of Minnesota, Columbia, and Brookhaven?
Well, first, DNA origami is a serious contender for building things below that 50-nanometer barrier, especially complex 3D shapes that traditional methods just can't handle. It gives us that needed precision.
Right. And second, they showed a practical way to integrate this DNA assembly with standard chip manufacturing. Using those gold pads and DNA anchors lets you build these things exactly where you want them, reliably over large areas. That hybrid approach is key.
Yeah, it makes it scalable. And third, they proved you can convert these DNA blueprints into actual functional electronic materials, like the tin oxide sensor. That huge surface area gives these devices potentially superior performance for things like sensors or maybe even catalysts or photonic devices.
So the really big picture here. DNA is stepping out of the biology lab and becoming a genuine engineering tool. We can program it to build not just life, but complex, functional, non-biological technology.
That's the potential, yeah. The authors themselves mentioned the next step is getting better controlled, not just vertically, but also how the crystals align horizontally on the chip, the XY plane control.
Yeah.
That would make manufacturing even simpler.
Which leads us to that final thought for you to chew on. If we can now use DNA, life's own blueprint, to program the construction of functional 3D electronics with this kind of precision, almost atom by atom, what's really stopping us from eventually building those hyper-complex AI systems or quantum computers we usually only see in science fiction? It feels like a fundamental tool just got added to the engineering workbench.